contain the peptidoglycan. Instead of that, they have the proteins and the lipids. But some archaebacteria, they shows the property of pseudopeptidoglycan. In that category of archaebacteria, instead of NAM, that is N-acetyl-muramic acid, it is being replaced by N-acetyl-talosamine uronic acid, and instead of L-amino acid, it has a D-amino acids. That is what a difference is in that. It is not in all cases of archaebacteria. In fact, we can say that all the archaebacteria doesn't show the same type of the structure. It is differing, as I have told, and sir has told about gram positive and gram negative. There is a variations in the archaebacteria because some lives at a high salt concentration, temperature, pressure. So there is a difference. They make them stable according to the environment. And another one difference which I have read in the previous slide, here instead of beta 1,4 linkage, they have beta 1,3 linkage. Okay, so that is what the difference in the cell wall of the archaebacteria. Next slides, please. And this type of properties you can see in some type of methanogenic microorganisms or halophilic microorganisms. As I told, they want to make them survive, they want to make them stable, so they change in their cell wall according to the need. Now this is what an overall picture of a gram positive and a gram negative cell. Now you can compare that. It is a simple one and another one is a complex one. Gram positive is a simple one, gram negative is a complex one.
So, as we discussed that we have compared the various structure and substructure of gram positive bacteria, gram negative bacteria, archaeal bacteria which are known as extremophile like methanogenic bacteria, halophilic bacteria, thermoacidophiles, and acid-fast bacteria. This all bacterial cell shows different pattern of their chemical nature. Though they have the same organelles, but if you see at the anatomy of that organelles, it shows some different chemical pattern. Now we know that for the classification point of view, nowadays we know that we are using the molecular techniques for the classification and categorizing bacterial cell. There was a method, and we are also practicing that method, that is known as chemotaxonomy. And this information, particularly the chemical pattern of different bacterial cell components, are useful, and it is very, very strong, and you can say the base on which we can rely. And as we already discussed, that certain group of chemical species are species characteristic of some selected bacteria, which is present in one bacterial cell, but it is not present in another bacterial cell. So this particular pattern of this chemical species we can take as a base for the identification and categorizing different bacterial communities. As we know that till date 1 to 2% bacteria are known and cultivated. We know that till 98 to 99% bacteria we are not able to cultivate in laboratory, and for which we are relying on the structure DNA, which organisms which are not cultivable in bacteria. A very good branch of emerging branch of science is coming, that is metagenomics, and that part is probably the future of various types of microorganisms.
Okay, thank you very much Dr. Ramesh and Dr. Chami. They have presented a very in-depth lecture regarding the cell wall of gram positive and gram negative bacteria. Now I would like to invite any questions from the students. If they have any doubt regarding this, we still have 10 minutes time where we can discuss these doubts of yours. So I encourage the students, if they have any doubts, they can come up with it. Meanwhile, I would also like to add 1 or 2 points. One thing which is very unique about this bacterial cell, as you have seen, that peptidoglycan or lipopolysaccharide, whatever the composition is, they are basically a very large macromolecules. Then the question arises how these macromolecules are synthesized outside the cell, or how these structures they are made outside the cell. But let me tell you one point in very brief, that basically these structures, they are not synthesized outside, they are just transported from the cytoplasmic molecule to the cell and where they are attached, where they are assembled. And for this, a very important role is played by certain carrier molecules like UDP, bacterial, which are responsible for taking these molecules. Synthesis of amino acids, synthesis of glucosamine structures, everything takes place inside the cell, that is the cytoplasmic pool. And with the help of these carrier molecules like bacterial, they are being transferred to the outer side of the cells, where the assembly takes place. So this is a highly intricate and complicated mechanisms which bacteria shows that even the large complex macromolecules can be made or can be used for the protection of the cells. Thus, to sum up, we can say that the cell wall is something like a very strong fortress which in old days kings used to build surrounding the city, where the public or the citizens would be protected from any invasions. Similar to that, these cell walls, they protect their vital components which are present in the cytoplasm. They perform not only this protection functions, but apart from that, as you have been listening, they provide the shape to the cells. They are also the site of phage attachment. Then they are also responsible for imparting a specific antigenicity to the cells. We call them as an O antigens. That is the somatic antigens, which is due to the presence of these outer membranes, the cell walls. Moreover, they also help in serological identification of the cells, because when we have to identify the organism, basically a microbiologist, if it doesn't get any clues, then he is a lost person, because there are innumerable number of microorganisms and he has devised a number of ways to identify it. As Dr. Ramesh said, that the chemical composition of the cell wall also plays an important role in identification. So not only serological identification, but there are also the identification made on the basis of phage attachment sites. Bacteriophages, they also have a particular preference for a particular site, particular chemical groups on which they will attach, and then they will carry out their penetration of nucleic acid inside the cell. Likewise, it also has other function which is performed, that is to prevent the loss of enzymes which are present in a periplasmic. Periplasmic space is full of enzymes which are responsible for ATP generation, which are important for various vital functions of the cells. Thus, in a way, you can say cell wall performs so many important functions that a cell cannot afford to lose it. However, in certain cases, as it has been earlier mentioned, that with the help of certain enzymes or in presence of certain antibiotics, we are able to make the cell wall, or we are able to lose the cell wall, or we are able to see that the cell wall is detached from the cell. But then under those conditions, the cell only survives if the solution and other things or the environment is isotonic, which does not permit the cell to shrink, nor it permits cells to burst. So these are in short various functions which have been explained to you in very detail by Dr. Ramesh and Dr. Chami. Dr. Ramesh, you have anything to add to these things?
I would like to add something.
Yeah,
last.
please go on.
We know that Gujarat is known for pharmaceutical industries. While we all are behind pharmaceutical industries, we know that pharmaceutical industries are producing a lot of molecules which target the cells. And as we know that there are four main targets of any medicine. Yeah. That is the inhibition of protein biosynthesis, inhibition of DNA replication, inhibition of cytoplasmic membrane, and you can say inhibition of cell wall biosynthesis. So the pharmaceutical industries are first of all targeting the first, you can say if a particular fort is there which is surrounded by four protective layer, So very first layer is cell wall. If you are able to break this cell wall, then everything is possible. Fine. So the cell wall is the structure. Knowing the detailed chemical structure of cell wall, pharmaceutical industry can devise novel target molecules which can reach to the pathogenic microorganism. We know that when pathogenic microorganism is invading, so if we start from the mouth to complete the entire intestinal tract, it contains more than 1000 type of microorganisms.
Correct.
And along with this, more than 1000 microorganisms or pathogenic microorganisms are also there. So knowing the detailed structure of cell wall, it will be useful for industries associated with pharmaceuticals to control the pathogenic microorganism particularly.
Yeah, Dr. Kothari very rightly said that if you break the first line of defense, then it is easier for organisms to, or for to control these organisms which are basically pathogenic. Therefore, the knowledge of cell wall is very important, not only for gram positive or gram negative organisms, but there are certain emerging diseases where the organisms, we have been able to look into those organisms for the first time. They may have certain different kinds of composition, and don't forget that organisms, they always have an ability to mutate, that is to bypass the various aggressive role of an antibiotic or a chemicals. So it is a constant fight between microorganisms as well as the scientists to control them by using various chemicals or drugs or medicines or antibiotics. Thank you very much, Dr. Ramesh and Dr. Chami. I am highly obliged to you people because you have accepted the invitation and come to the studio to deliver a lecture. I know it is very difficult to prepare and spend such a lengthy lecture in such a perfect manner. So I personally thank you, as well as I would also like to thank the Director Higher Education who has initiated this program so that the live lectures reach to the different students across the Gujarat for various subjects. I am also thankful to Dr. Kamal, who has taken initiative to carry out and coordinate the BISEC lectures, SANDHAN lectures. And last but not the least, I am also very much thankful to the various crews, the staff of BISEC studio, who have remained behind the curtain but have been able to bring the excellent quality of this live coverage for the students. Thank you very much. Namaste.